Assalamu alaikum wa rahmatullahi wa barakatuh. Today we will continue with activity 1 of chapter 3, Proteins and Association of Amino Acids. At the end of our lesson for today, we will be able to write the chemical equation of the reaction of a dipeptide formation using general formulas. List the characteristics of the protein, the number of amino acids, their sequence, and the number of each amino acid. List the different structures of proteins, differentiating among them the primary, secondary, tertiary, and quaternary levels. Define denaturation and classify proteins into functional and structural proteins. You can refer to the summary sheet that you are supplied with to revise the required information. First, let's recall back the characteristics of a protein molecule. Every protein has a specific number of amino acids, specific number of each type of amino acids, and a specific sequence of amino acids. Now, amino acids form up a protein molecule. But how is this peptide chain formed? Now we'll discuss dipeptide formation. First of all, let's define a dipeptide. A dipeptide is a molecule made up of two amino acids joined together. Dipeptide formation is a synthesis reaction. The reaction between two amino acids occurs between the hydroxyl group of the first amino acid and the hydrogen atom of the amino group of the second amino acid. Their bonding together gives a water molecule, and the two amino acids will be joined together by the peptide bond as the following reaction. This corresponds to the peptide bond between the two amino acids and the resulting water molecule. Now, what if the protein molecule is to be broken down? How does this occur? The breaking down of proteins is called hydrolysis. Hydrolysis is the reverse reaction of synthesis, and it occurs in the presence of water. A dipeptide is broken down in the presence of water. The peptide bond will be broken down, and the water molecule will be dissociated into two parts, the hydroxyl group and hydrogen atom. Hydroxyl group will associate with the carbon of the first amino acid to form carboxylic group, and the hydrogen atom will associate with the amino group of the second amino acid. What happens after the protein is produced? After it is produced, the protein would do its function directly, or it would be produced in a non-functional way and then it gains its function, or maybe the produced protein would be subjected to certain factors that makes it lose its function. Then what characteristics make the protein functional? Let's take the example of insulin. Insulin is a hormone that plays an essential role in the regulation of glycemia, that is the glucose level in blood. Insulin first is produced in the form of proinsulin, having three chains, A chain, B chain, and C chain, before it turns into insulin having only A chain and B chain. Then the difference between the two molecules is the presence of C chain in proinsulin and its absence in insulin molecules. Then, the first factor that determines the functioning of a protein is the sequence of amino acids. Let's study another example, ribonuclease. It is an enzyme that degrades RNA. This protein loses its function when it changes from the folded state into unfolded state under the effect of denaturing agents that are urea and mercaptoethanol. This image shows the folded state of the ribonuclease, that is when subjected to the denaturing agents urea and mercaptoethanol, it turns to be unfolded and becoming inactive. Now the second factor that we can come up here with is the configuration of the peptide sequence. Its configuration affects the functioning of a protein. That is, when it is in a certain given structure or form, it would be either functional or non-functional. To sum up, the function of a protein depends on the sequence of amino acids and the three-dimensional structure of the protein. After a peptide chain is formed, certain changes occur before the protein reaches its final structure. The following video shows the different levels of the protein structures.
The primary structure of the protein is the linear sequence of amino acids as encoded by DNA. The amino acids are joined by peptide bonds, which link an amino group and a carboxyl group. A water molecule is released each time a bond is formed. Specific amino acid sequences give proteins their distinct shapes and chemical characteristics. These protein chains often fold into two types of secondary structures stabilized by hydrogen bonds. A protein chain can fold into a rigid alpha helix, forming regular patterns of hydrogen bonds between the backbone atoms of nearby amino acids. Backbone atoms of the chain can interact side by side to form beta sheets. Many proteins fold into a compact globular shape, with hydrophobic side chains sheltered inside away from the surrounding water. The functions of many proteins rely on this folded structure. For instance, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds oxygen. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so that the complex can pick up more oxygen in the lungs and release it in the body.
Based on the previous animation, how many levels of a protein structure are there? There are 4 levels: the primary, secondary, tertiary, and quaternary. As we've explained before, certain proteins lose their function or they become inactive after certain modifications or changes in their structure. Then how can a protein undergo these modifications in the structure? This actually occurs due to the destruction of the bonds that keep the configuration stable. This is described as denaturation of proteins, that is changing the nature or the 3D structure of the protein molecule. Denaturation of proteins is the disruption and possible destruction of both the secondary and tertiary structures, but not the primary. After the denaturation of proteins, the primary level remains stable, unchanged. This is because the denaturation reactions are not strong enough to break the peptide bonds. Now we'll talk about the classification of proteins. Proteins can be either structural, that is, they form up the structure of the cell, or functional, they provide different functions at the cellular and systemic levels. Structural proteins in the body, like collagen in the skin and keratin in the hair, while functional proteins are like enzymes and hormones. To sum up, a dipeptide is a molecule made up of 2 amino acids joined together. Dipeptide formation occurs according to a synthesis reaction, and a peptide is decomposed according to a hydrolysis reaction. The carboxyl group of the first amino acid associates with the hydrogen atom of the amino group of the second amino acid to give water molecule. This is synthesis reaction, where a peptide molecule is formed. While hydrolysis reaction occurs in the inverse direction, where the peptide molecule breaks down in the presence of water molecule to give 2 separate amino acids. Proteins have different levels in their structure. Primary, corresponding to the sequence of amino acids in it. Secondary, corresponding to its fold. Tertiary, corresponding to its spatial configuration, 3D structure. And quaternary, it is the case where proteins have more than one polypeptide chains. Denaturation is the disruption and possible destruction of both the secondary and tertiary structures of the protein, but not the primary. And finally, proteins can be classified into functional, having specific functions, and structural proteins that form up the cell's structure. Thanks for listening.